Imagine standing face to face with a creature that roamed the earth over 150 million years ago. A towering Brachiosaurus or the fearsome Tyrannosaurus rex. These species ruled a world long gone, yet today advances in genetic science are turning this science fiction into a potential scientific frontier. Good evening, everyone present here. Today, myself, Oshani Ghosh, along with my teammates, are going to conduct episode 2 of The Last Brain Sale Extinct Not Forever. Welcome to our exploration of de extinction, the fascinating concept of reviving extinct species, with a focus on the giants of the Jurassic world. From ancient DNA extraction to cloning and genome editing, we'll walk through the cutting edge technologies and ethical questions behind this bold scientific endeavor. First of all, what is de extinction? De-extinction is the process of bringing back extinct species through advanced biotechnologies like cloning, gene editing, and synthetic biology. It's not just about dinosaurs, it's about reviving species like the woolly mammoth or the passenger pigeon. This is not science fiction anymore, it is science in action. Let's start with the why. Species are going extinct at an alarming rate, around 1,000 times the natural background rate, according to scientists. Some extinctions are natural, but many are human driven. Habitat loss, pollution, hunting, and climate change. De-extinction offers the several potential benefits. Ecological restoration, bringing back keystone species can help restore damaged ecosystems. Scientific insight. Studying revived species can teach us about evolution and genetics. Ethical responsibility. Some argue that if we caused extinction, we should at least try to reverse it. But how? How can we possibly bring back something that is gone? Let's delve into the core scientific techniques involved. First, cloning. Cloning was first made famous by Dolly the sheep in 1996. Cloning, specifically through somatic cell nuclear transfer (SCNT), is a technique where the nucleus of a somatic body cell from an extinct species is transferred into an egg cell of a closely related species that has had its nucleus removed. Now, coming to the steps. First, DNA extraction from extinct organisms. Scientists extract DNA from preserved tissues, frozen specimens, museum samples, or desiccated remains. The quality of DNA is crucial. Cloning typically requires relatively intact nuclear DNA, which limits the method to recently extinct species. Example 20 to 50 years old. Collection of enucleated egg cells. An egg is taken from a closely related extinct species, example domestic goat for an extinct ibex. The egg's own nucleus is carefully removed. This is called enucleation. Now, nuclear transfer. The nucleus of the extinct species cell is inserted into the enucleated egg. This essentially reprograms the egg cell to act like a fertilized zygote with the extinct animal's DNA. Then embryonic development, the hybrid egg is stimulated to start dividing as an embryo. Early stage development is monitored in vitro in a lab dish for viability. Now, implantation into surrogate. A viable embryo is implanted into the uterus of a surrogate female of the extant species. The surrogate carries the fetus to term. Then birth and growth. If successful, the offspring will be a clone of the extinct species or as close as biologically possible. This method was used to attempt the revival of the Pyrenean ibex in 2003. The clone survived for just 7 minutes. Similar method was used for the extinction of dire wolf, which is widely recognized as the first species to be successfully de-extincted. Now, DNA decays over time. For long extinct animals like dinosaurs, this method can be a challenge. Now coming to genome editing, CRISPR-Cas9 technology. Genome editing is the process of making precise, targeted changes to an organism's DNA. The most widely used tool today is CRISPR-Cas9, which acts like molecular scissors to cut and modify DNA at specific locations. Now, how genome editing works in de-extinction. Rather than cloning an entire extinct genome, genome editing allows scientists to engineer the genome of a closely related living species to include traits of the extinct one. First, identify the extinct species genome. Scientists sequence the DNA of the extinct organism using samples, example bones, skin. They compare it with the genome of a close living relative. Second, find target genes. Using bioinformatics, they locate specific genes responsible for key traits like cold resistance, thick fur. These genes are then isolated and cataloged for editing. Third, design CRISPR tools. Scientists create a guide RNA that leads the Cas9 protein to the exact place in the genome of the living species. Cas9 then cuts the DNA, allowing insertion or replacement of specific extinct genes. Fourth, edit the embryo or cell. The edited DNA is introduced into the embryonic cell or induced pluripotent stem cells (iPSCs) of the living species. Fifth, generate embryo and implant. The edited cells are grown into embryos and implanted into a surrogate. The resulting organism carries traits of the extinct species like a genetic hybrid. Now, coming to example. For example, the woolly mammoth is closely related to the Asian elephant. Scientists identify genes in mammoth DNA like those for cold resistance. They use CRISPR to insert these genes into the elephant genome. The resulting hybrid could be grown in a lab or implanted in a surrogate to create a mammoth plant. Now, scientists have successfully inserted mammoth genes into elephant cells. The next step is embryo creation and artificial womb development. Third, now coming to backbreeding. What is backbreeding? Backbreeding is a selective breeding technique used to recreate an extinct species or a close approximation by identifying and breeding individuals of a related living species that still carry traits of the extinct one. It doesn't bring back the original genetic identity of an extinct animal, but attempts to reconstruct its appearance, behavior, and ecological role through repeated breeding over generations. How it works: first, identify the extinct species traits. Researchers study fossil records, written descriptions, illustrations, and preserved specimens to determine key traits like size, color, horn shape, behavior, etc. Second, find descendant or related species. Scientists look for living species that still retain some of those ancient traits. Third, selective breeding. Individuals with the most desired traits are bred, their offspring are again selectively bred, and this process continues for many generations. The result is, after several generations, often 10 plus, the offspring may strongly resemble the extinct species in terms of appearance and ideally ecological function. For example, take the Orosh, the wild ancestor of domestic cattle. It went extinct in the 1600s. Scientists are cross-breeding modern cattle that have Oroshes-like traits to recreate something visually and genetically close. Thus, de-extinction is no longer just the realm of science fiction novels. It's a real, complex, and rapidly advancing field that brings together genetics, conservation, biology, and biotechnology. Thank you.
Hello everyone, this is Akshara. Now, the first thing when you think about bringing back extinct species, the first thing which comes to your mind is colossal biosciences. Colossal biosciences was found by Harvard geneticist George Church and tech entrepreneur Ben Lamb, based in Dallas, Texas. Colossal is pioneering the intersection of de extinction, genetic engineering, and ecosystem restoration at an unprecedented scale. Next, please. Now, why is Colossal Biosciences doing it? They aim to bring functionally similar versions of extinct species to restore disrupted ecosystem. It is a private biotech de-extinction company with around 175 scientists focused on reviving extinct species like mammoth, dodo, thylacine, direwolf, and genetic rescue and conservation genomics. Next. Now, woolly mammoth. Colossal Sciences is not actually cloning a mammoth. That's impossible. DNA degrades after death, and mammoths have been extinct for over 4,000 years. So, why do I think we are doing it? To basically help fight climate change. The Arctic is warming, and frozen ground called permafrost is starting to melt. When it does, it releases gases like CO2, methane that make global warming worse. Mammoths once helped keep the ground cold by trampling snow so cold air could reach the ground. Colossal believes bringing back mammoth like animals could slow the melt and help protect the climate. Now, as Oshani mentioned, how CRISPR-Cas9 helps bringing back the woolly mammoth species, we basically scientists extract DNA from mammoth bones found frozen in Siberia. They then figure out which parts of the mammoth genome make it cold resistant. Using CRISPR, they insert these mammoth traits into elephant cells. The edited cells are then turned into embryos and possibly grown in lab made wombs or in elephant surrogates. Colossal hopes to have its first mammoth elephant hybrid calf born by 2028. In 2025, they made a woolly mouse, a mouse with mammoth traits like cold resistant, fat, and fuzzy fur, which was a major breakthrough. Next. Tasmanian tiger. The Tasmanian tiger, or thylacine, was a unique carnivorous marsupial native to Australia and Tasmania. Officially declared extinct in the 20th century, the species has become a symbol of lost biodiversity. Despite its dog like appearance, the thylacine was a marsupial more closely related to kangaroos than wolves uh, or dogs. Now, why do we need to bring back Tasmanian tiger? To restore balance in ecosystems. The Tasmanian tiger was a top predator in Australia and Tasmania until it went extinct in the 1930s. Without it, the local ecosystems became unbalanced. Some animal populations grew, others declined. They believe that reviving the thylacine could help fix damaged ecosystems in places like Tasmania and Australia. Now, how do we do it? The Tasmanian tiger's closest relative is the fat tailed dunnet, a tiny mouse sized marsupial. The scientists have sequenced the DNA from preserved thylacine specimens, which are found in the Victoria Museum, Australia. They then compare the DNA to the donor DNA, and then they see what it needs to change. Using CRISPR to edit the donor's genome into a thylacine-like one. They then turn the edited DNA into embryos in the lab. The embryos are then implanted into a donor or artificial womb is used. If this works, they raise the animal in controlled habitats and eventually release it to the wild. Colossal and the University of Melbourne have sequenced a 99.9% complete thylacine genome from museum specimens, a first of its kind achievement for an extinct species. Next. Dodo. Now, everyone has heard of Dodo. And why do you think it's very famous? Because Dodo went extinct shockingly fast. The dodos lived peacefully on the island of Mauritius with no natural predators. But when humans arrived in the 1600s, they brought rats, pigs, monkeys that ate dodos' eggs. In just about 60 years, the dodo was completely wiped out. It became one of the first animals to go extinct because of humans. Colossal aims to bring the dodo back not by cloning but by editing the genome of its closest living relative, the Nicobar pigeon, then using birds like chickens as surrogate parents. Now, how do we actually do it? The sequenced fully genomes of the dodo, its cousin solitaire, and the Nicobar pigeons. We use AI to find the gene for traits like beak shape and body size. The edited cells are then injected into embryos. Chimeric chicks are successfully hatched. The partnered with the Mauritian Wildlife Foundation to restore habitats and eventually release the dodo. Next. The dire wolf. This dire wolf was the most famous in the recent months. In October 2024 and January 2025, Colossal welcomed three puppies, Romulus, Remus, and Khaleesi via gene-edited grey wolf embryos implanted in dog surrogate. Colossal frames it as functional de extension, meaning these animals can fill the same ecological niche even if they aren't perfect genetic replicas. But there are many paleontologists, scientists, and biologists who say that they aren't true dire wolves. They're just grey wolves with dire-like traits, and are meant to play similar role, but they aren't meant to play a similar role in nature. Now, why was the dire wolf made? To test de-extinction technology. The Direwolf project is a tech test run to push the limits of gene editing, cloning, and synthetic biology in large mammals. The Direwolf project is Colossal's first successful live outcome. Previous milestones like the woolly mouse were important groundwork, but no animals were born. Thank you.
Hi everyone, I'm Aditi. I'll be talking about the ecological and environmental impact as well as some of the ethical concerns. These species acted as ecosystem engineers and keystone species, helping to restore balance, boost biodiversity, and even mitigate climate change. But on the other hand, reintroducing them into today's ecosystems comes with risks, like unpredictable impacts on existing wildlife and habitats. Let's begin with the woolly mammoth. These ancient giants were more than just ice age icons. They were ecosystem engineers. Through grazing, trampling, and foraging, mammoths suppressed tree and shrub growth and transformed mossy tundra into vast open grasslands. This process, known as the restoration of the mammoth steppe, could be key to stabilizing the Arctic. By converting forest and tundra back into grassland, mammoths would help preserve permafrost and support a reflective carbon sequestering landscape. While they wouldn't increase ice coverage, their return could play a crucial role in slowing climate change. Now let's talk about aurochs, the wild ancestors of domestic cattle. Aurochs were keystone grazers. Their movements created open and semi-open habitats and promoted a rich mosaic of grasslands, shrublands, and woodlands. Their grazing patterns left gaps in vegetation, allowing wildflowers and pioneer plants to thrive. Their dung fertilized the soil, and their trampling disturbed it just enough to allow new plants to take root. Birds used their fur for nesting, and most importantly, smaller herbivores found safety within their herds. Mm, And finally, considering the passenger pigeon, once numbering in the billions, these birds were a force of nature in Northern American forests. Their massive flocks opened up forest canopies, cycled nutrients, and enriched soils. Reviving the passenger pigeon could help restore these ecological functions and enhance the health of our forests. De extinction is often considered a double edged sword, even though it presents an impressive solution to the biodiversity loss being faced today. It comes with a mammoth of risks. Reviving extinct species risks resurrecting ancient pathogens, organisms our immune system and ecosystems are no longer prepared for. There's also the threat of ecosystem disruption, where reintroduced species may outcompete or destabilize native populations. A habitat mismatch is likely, as modern ecosystems have changed drastically since these species last existed. Finally, resource diversion. The allocation of funds and attention away from protecting existing endangered species raises conservation concerns. A measured, evidence based approach is essential before taking these irreversible steps. The ethics of de extinction involve complex scientific, legal, and philosophical considerations. One major issue is objectification, the idea that de extinct animals may be treated as bio-objects, entities controlled, engineered, and manipulated solely for human benefit. This reduction of living beings to tools or experiments risks ignoring their intrinsic dignity and welfare. Next comes the risks involved while using artificial wombs, for example, using elephants to gestate woolly mammoth embryos. But these surrogate mothers face serious concerns, invasive procedures, physiological stress, and emotional or cognitive suffering. And what about the animals we bring back? They may enter a world for which they are no longer suited, facing habitat mismatch, social isolation, or the absence of species specific learning, especially if raised without other of their kind. Many may suffer from genetic instability, health issues, or psychological stress when confined to artificial environments. The legal and philosophical aspects include species classification and legal status, patentability and ownership, and the biggest question is it okay for humans to play God? I would like to end with my thought. I would like to end my thoughts on de-extinction. I believe the full potential of this technology for conservation may not be achieved unless issues such as overhunting and deforestation are also addressed. Thank you.
Hey guys, it's Aditya, it's me again. And well, here we are for the Jurassic style part of the presentation. Next slide, please. Now, I know that it's called Jurassic style, but I feel like we have to give proper attention to the other eras, which are the Cretaceous era and the Triassic era, because all of them are branded using the term Jurassic. However, we often neglect the complete geological time scale. Next slide, please. Okay, so here you can see it's a pretty colorful and illustrative example of the main major eras, which are the Mesozoic era, Cenozoic era, Paleozoic era, Proterozoic era, and Archean. Archean and Proterozoic are more for geology because there was not no life back then. The earliest forms of life were Paleozoic, and then Mesozoic is the part which we find interesting, which contains the Triassic, Jurassic, and Cretaceous part. So I find it very incorrect to always label it as Jurassic. Next slide, please. So this is just a the fun diagram to show you that which species were in which eras. In fact, the most famous of them all, the T-Rex or the most famous Triceratops or the Parasaurs, all of them are from the Cretaceous period. The Jurassic period was more herbivore dominant, as you can clearly see here. Next slide, please. So, why aren't we able to perform our de-extinction efforts on these species? Well, quite simply speaking, the half life of DNA is pretty less. It's around 517 years. So, it's been more than 66 million years since our reptilian friends have been alive, and hence we do not have any sort of DNA which is viable enough for studies. Hence, even though we have found a lot of intact fossils, none of the DNA is close enough. And believe it or not, the closest living relatives to dinosaurs are birds. Your chicken is more related to the dinosaur than larger animals. So there's no proper surrogate as well. So birds are modern dinosaurs. Yes, they are. Chickens, ostriches, and cassowaries are descendants. In fact, the chicken is the probably genetically the most close to current. And there was a theoretical project called Chickenosaurus, which was aimed to reverse engineer DNA. However, it was not possible. There was no surrogate womb. And even if we did somehow have DNA, as earlier my teammates mentioned, that the somatic cells have to be from a very closely related species. So we don't have any compatible species to gestate this like we do for the woolly mammoth or the dire wolf. Next slide, please. So, is it possible in the future? Well, theoretically, it is. If we are able to make such grand advancements in synthetic biology that we are able to completely make a genome based on prior data, or maybe even with the use of agentic or generative AI, then maybe in a couple decades or 100 years, and if we are able to completely create artificial wombs, have completely in vitro wombs which are eliminating the need for a surrogate, then also if we have a genome, it's possible. And of course, even having a genome, we need to have advanced gene editing. We need updated version of CRISPR. The current version of CRISPR is definitely not reverse engineering. Next slide, please. So now the main issue here is, uh, let's say we do overcome all those hurdles magically. The ecological impact, environmental challenges, and ethical questions, the three E's are very important. I think Aditi already covered this in great detail, but the ecological impact, it's massive disruption. I mean, where are you going to keep your brachiosaurus in your backyard? It's not possible. This is you cannot open the fridge, put the dinosaur, and close the fridge again, unfortunately. And again, invasive species, dinosaurs in current lifestyles, how we do not know how they will interact with other species, and whether they would interact well with their own, rather. And of course, any sort of mesotic mega viruses which could take down herds and herds of dinosaurs. We definitely do not want that in our human environments. COVID was already bad enough, and it's apparently on the rise again. So definitely, the disease transmission is not the ideal part of it. There are of course environmental challenges. The food chain is absolutely disrupted. I mean, we consider human beings on the top of the food chain. I'm not sure what the T-Rex has to say for that. And then there's the climate mismatch. The climate during the Mesozoic era was completely different. Forget Mesozoic era. There were such high variations in how the Earth was in the Cretaceous, in the Jurassic, and the Triassic. In fact, the Cretaceous period was so diverse that they were divided further into late, early, and median, because Earth was in such a disruptive stage that time. And urban risk, of course, we've watched the movies. I don't know where we are going to be traveling with these big creatures. Definitely not suitable for an urban environment. And of course, can be released in the wild as well. Now, there's some ethical questions which are up for debate, whether it's right or not, or whether it's safe or not, or whether it's important or not. I personally would love to be involved in a project like this, but what is it really doing for the biological community except satisfying our needs? Next slide, please. So, before we sign off, I want to thank my team members, because even though I spoke about the title part, they have put the highlight on the actual science of it, the actual research and the actual methodology, which are important for us as biotechnology students. So, a virtual round of applause for them, please. And before you end this meeting, I know you guys won't say anything if I ask you to unmute it. Please drop down your favorite dinosaur in the chat box. It'd be really appreciated. Thank you all for tuning in the Jurassic style of this last brain cell, and it was fun.